Welcome to this lecture on the properties and functions of cytokines. Imagine managing a massive construction site with thousands of workers, but none of them have cell phones or walkie-talkies. It would be absolute chaos. In your body, the immune system faces a similar challenge, with millions of white blood cells scattered everywhere. Today, we are going to learn exactly how these cells communicate, coordinate, and execute their defensive strategies to keep you healthy. To understand why cytokines are so important, we first need to look at the problem they solve. When a virus invades your body, the immune cells that detect it need a way to call for backup. Because cells cannot speak, they use chemical signals. Cytokines act as the biological equivalent of text messages or emails. By releasing these chemical messages into the surrounding fluid, a single cell can instantly alert the rest of the immune system, telling other cells exactly where to go, how to attack the invader, and just as importantly, when to calm down once the threat is neutralized. Now that we know why they are needed, let us define what they actually are. In formal immunological terms, cytokines are low molecular weight soluble proteins, or glycoproteins. Unlike hormones, which are produced by centralized glands, cytokines are secreted by a wide variety of immune cells, primarily helper T cells and macrophages. Their main job is to mediate and regulate immunity, inflammation, and hematopoiesis, which is the production of new blood cells. When a cytokine reaches a target cell, it triggers a cascade of internal signals that alters the cell's gene expression, instructing it to grow, differentiate, or even undergo programmed cell death. Understanding what cytokines are naturally leads us to how they travel. Cytokines operate through three main functional pathways. The first is autocrine signaling, where the cell that releases the cytokine also responds to it, essentially leaving a note for itself to stay activated. The second is paracrine signaling. Here, the cytokine travels a short distance to activate a neighboring cell, which is the most common way cells collaborate in a lymph node. Finally, in rare cases, cytokines use endocrine signaling. They travel through the bloodstream to affect distant organs, such as when they tell the brain to induce a fever. Beyond their travel pathways, cytokines exhibit unique behavioral properties that make our immune system incredibly robust. First is pleiotropy, meaning a single cytokine can have completely different effects depending on which cell it hits. Second is redundancy, where multiple different cytokines can trigger the exact same function, ensuring that if one gene fails, the immune system does not collapse. Cytokines can also work in synergy, combining forces to produce a massive effect. Or they can display antagonism, blocking each other's actions. Without these overlapping properties, our immune system would either be too weak to fight a cold or so aggressive it would destroy our own organs. These complex behaviors bring us to a crucial question. How do cells actually receive these signals? Cytokines do not just float inside cells. They must physically lock into specific receptor proteins on the outside of the target cell's membrane. The effectiveness of a cytokine signal depends entirely on how many of these receptors are currently occupied by cytokine molecules. Because these receptors have such a high affinity for their specific cytokines, they can trigger massive cellular changes, even when the concentration of cytokines in the fluid is incredibly low. To truly understand this, we need to look at the mathematics behind it. To model this receptor binding mathematically, we use the fractional occupancy equation. This formula comes from the principles of biochemistry and describes the relationship between the cytokine concentration and the percentage of activated receptors. In this equation, Y represents the fractional occupancy, which is the fraction of receptors bound to a cytokine. The variable C represents the concentration of the cytokine in the surrounding fluid, measured in molarity. Finally, Kd is the dissociation constant. This measures the receptor's affinity for the cytokine. If Kd decreases, it means the receptor grabs the cytokine more tightly, so the cell requires a much lower concentration of cytokines to become fully activated. Now that we have our formula, let us see this with a concrete example. A helper T cell secretes interleukin-2, and the local concentration is measured to be 10 to the negative 10 molar. The receptor's dissociation constant is 10 to the negative 11 molar. We want to find the fractional occupancy, Y. We substitute our given values into the equation. To make the addition easier, we use a standard algebraic trick. We express the cytokine concentration as 10 times 10 to the negative 11. This allows us to cancel out the powers of 10. We are left with 10 divided by 11, which gives approximately 0.909. Physically, this means 91% of the receptors are bound, sending a massive signal to the cell. Let us look at another scenario, this time working backward. A macrophage needs 50% receptor occupancy to activate, so Y equals 0.5. The dissociation constant is 2 times 10 to the negative 10 molar. We need to find the required cytokine concentration, C. We plug our known values into the formula. To solve for C, we multiply both sides by the denominator to remove the fraction. Next, we distribute the 0.5 into the parentheses, which simplifies the constant term beautifully. We then subtract 0.5C from both sides to group our variables. Finally, dividing by 0.5 gives us our answer. The required concentration is exactly 2 times 10 to the negative 10 molar. Notice something interesting from that last example. When the required occupancy was exactly 50%, the required concentration perfectly matched the dissociation constant. Let us prove that this is not a coincidence, but a fundamental rule of receptor biology. We're setting out to prove the exact physical meaning of the dissociation constant. We start with our standard fractional occupancy equation. We want to find the general relationship when exactly half of the receptors are occupied, so we set Y to 0.5. To make the algebra cleaner for our proof, we will rewrite 0.5 as the fraction 1/2. This sets up a simple proportion that we can solve. Continuing our derivation, we cross-multiply the fractions. 1 times the denominator gives us Kd plus C, and 2 times the numerator gives us 2C. This leaves us with a very simple linear equation. We subtract C from both sides to isolate Kd. The result is Kd equals C. We have just proven a fundamental rule of biochemistry. The dissociation constant is physically defined as the exact concentration of ligand needed to occupy half of the available receptors. This is why Kd is the perfect measure of binding affinity. A lower Kd means you need fewer cytokines to reach that 50% activation threshold. Now that we have proven the math, let us visualize these concepts together. On the left side of this diagram, you can see pleiotropy in action. A single helper T cell is releasing cytokines that bind to 3 completely different cells, a B cell, a macrophage, and a cytotoxic T cell, causing different effects in each. On the top right, we see the physical receptor-cytokine complex on the cell membrane. Finally, look at the graph on the bottom right. The curve shows fractional occupancy rising as cytokine concentration increases, eventually maxing out at 1.0. Notice the dashed line. It perfectly illustrates our proof. Exactly at the 50% occupancy mark, the concentration on the horizontal axis is equal to KD. We have covered a lot of ground today, so let us summarize the most important ideas. First, cytokines are the essential chemical messengers that allow the immune system to communicate and execute its defensive strategies. Second, they are incredibly flexible, operating through multiple pathways and utilizing properties like pleiotropy and redundancy to ensure our immune response is fail-safe. Finally, we learned that cytokine communication is driven by high-affinity receptors. By mastering the fractional occupancy equation, we can mathematically determine exactly how much cytokine is needed to trigger an immune response. Understanding these principles is the foundation for treating severe immune disorders. Great job following along with the math and concepts today.